Hi, everybody. I'm Eva. Thank you for this nice introduction. I'm currently doing a postdoc at Scripps in San Diego, where I'm looking at how marine microbes interact. However, today I'm going to talk about one of my chapters in my PhD thesis, where I studied how environmental fluctuation and biotic interactions shape plankton community dynamics. And today I'm going to talk about one of my projects where I studied how fluctuation in nutrient supply and warming affect ecological networks in 10 Swiss lakes. So human impacts such as climate change and nutrient fluctuations are affecting aquatic ecosystems such as Lake Reifensee that you can see here on the picture around the world. A recent study just published that a 4-degree increase in temperature might have the same devastating consequences on lake water quality than the eutrophication of the 20th century. But who am I telling this? I think we saw during this conference that it's very clear that humans affect ecosystems or aquatic ecosystems. However, I think we still need to better understand how changes to one part in the ecosystem affect other parts and relative processes. And the best example for this is biotic interactions. So biotic interactions that you can see here, like predator-prey or also competition, are to me the engine of community dynamics, or like the centerpiece. And I think they perhaps remind the most overlooked component of biodiversity change. Biotic interactions, they can be summarized in networks. And such networks have proven to be fundamental to study large-scale changes in ecosystems. However, how entire networks will rewire as a consequence of human influences remains very scarce, just because we're missing long-term, well-created time series of complete ecological networks. And when they're available, they're often nonlinear in nature, and we need specific tools to, for example, infer interactions. Moreover, a lot of studies, they focus on only one type of interactions, for example, food web dynamics or competition. And then interactions have often been assumed to be constant, whereas recent studies showed that they might be actually changing. And then last, we might neglect potential interactions among drivers. So that's why in my PhD thesis, or in one of my chapters, I wanted to study the effects of two major anthropogenic stressors, which are nutrient fluctuation and warming on ecological networks in lakes. And to do so, I relied on 20 to 43 years of plankton abundance data. So plankton, besides being very charismatic organisms, as you can see here, they're the base of the aquatic food web. So higher organisms that we care for, like mammals or fish, they highly depend on them. Moreover, they're a global player in the global carbon cycle. And for example, they produce 50% of Earth's oxygen, which is like the same as all the trees and plants taken together. Plankton, they react very fast to changes in their environment, making them ideal target to see how the environment influences aquatic communities. And moreover, due to their fast generation times, they have been an ideal target for lake monitoring. So it means that if we can understand changes in plankton communities, we can also understand changes in the whole ecosystem and also ecosystem services. So plankton have traditionally been sampled weekly, monthly, or yearly. And the picture you can see here is from 100 years ago, taken in Lake Lucerne. And that's how plankton were traditionally monitored back then. And to be honest, not a lot has changed. So we still sample plankton with very similar methods. And this also means sampling organisms with a generation time of 15 days at a monthly basis is like the same of going to a small forest patch every 400 years. So this kind of data is very useful to understand long-term changes in communities. So the plankton data that I had came from 10 different Swiss lakes that were in the peri-alpine region. And so those lakes, since the 1970s, they underwent managed reoligoification, which is just the control of the release of phosphorus in the system by, for example, banishing phosphate from laundry detergents or just installing wastewater treatment plants. And phosphate is the main limiting factor for primary producer growth in those lakes. At the same time, the water temperature steadily increased while becoming exponential in the past 10-15 years, starting in the year 2010. And in the lakes, we observed an increase in water temperature from 0.4 to 1.7 degrees Celsius, which is pretty scary. This is like more than the average predicted by NASA of 1 degree since like the pre-industrial age. And so now, to understand how those two processes affect ecological networks in lakes, I grouped the zoo and phytoplankton taxa present in the lakes into well-known trophic guilds based on feeding behavior, body size, and nutritional requirement. And I did this to understand processes at the network level, but also to avoid potential misclassification and be able to compare better across those 10 lakes. So this resulted in a network of up to 16 nodes that you can see here. So this network encompassed invertebrate predators, omnivores, large herbivores, small grazers such as ciliates and rotifers, mixotrophic dinoflagellates, and primary producers. Whereas most of the guilds of primary producers I could divide into 2 nodes based on cell size and coloniality. All links in this networks were bidirectional, which also means they can go up and down the food web, allowing the study of trophic controls. So associated to each of those nodes is a time series of monthly abundance. Time series, they contain essential information about how one node affects another node. It is important to note that those time series were collected in the field, where a suite of biotic and abiotic variables are present, and making the relationship between those nodes potentially nonlinear, which this also means that statistical approaches that are based on correlation alone that we can use in controls experiment are not suitable here. So non-parametric approaches such as empirical dynamic modeling, they offer a promising way forward to work with such nonlinear, highly complex, chaotic system as plankton. And to then actually reconstruct the networks in those lakes, I used convergent cross-mapping. Convergent cross-mapping is very simply said a nonlinear causality test. And it quantifies the amount of shared information between 2 time series. Or like how the state of one node can be used to predict the state of another node. And the amount of shared information is called the cross-map accuracy, going from 0 to 1. And so by calculating this cross-map accuracy within moving windows of 16 months, and then comparing this to a seasonal surrogate null model, I could then track how connectance and interaction strength in those lakes changed over time. And connectance and interaction strength are very useful because they have been proven to show changes in the whole ecosystem, and there are a lot of studies relating them to ecosystem stability or also the maintenance of biodiversity. So the first question I want to ask is just, does connectance and interaction strength even change over time? And based on previous studies, I would expect that yes. So here on the first graph on the y-axis, you see the network connectance, which is just the amount of realized links at a given time point. And then the different lines, they represent the 10 lakes that we have. And as you can see here, the connectance is highly dynamic, so it changes over time. We see that maybe through reoligoification, there's a slight increase in connectance. Whereas when nutrients remain stable, it's somewhat mixed. So some increase, some lakes increase, some lakes decrease. But then if we focus on the warming period, we see that connectance in almost all the lakes is decreasing. And especially in Lake Zurich, which is here in dark blue. So the second graph on the y-axis shows you the average interaction strength, which is defined by this cross-map accuracy. And also here you can see that it is changing over time, but it seems to be less dynamic than connectance. And Lake Zurich here seems to have the weakest interaction. So now after seeing that they change over time, a logical follow-up question would just be to see if there's any relationship with the changes in water temperature and nutrient levels that we saw on the slides before. And to investigate these questions, I used S-maps, which are also from the empirical dynamic modeling framework, and they are locally weighted linear regressions that give points closer in state space a higher weight. And so I used this model to model network connectance and interaction strength as a function of water temperature, phosphate levels, and the lake's depth and volume to account for differences among the lake. And then I used this model to just predict network connectance and interaction strength over a gradient of temperature and phosphate levels. And this resulted then in the following plot. So here on the y-axis you have water temperature, and on the x-axis phosphate levels, and they go from the smallest to the highest observed value in the lake. And here I'm going to show you only the predictions for Lake Zurich, since Lake Zurich is a medium-sized lake and it's economically very important, for example, for fisheries, but also for the drinking water supply of the city of Zurich. And moreover, it's the lake we have the most consistent record, so those 42 years of data encompassing real liquefaction and the warming phase. So the color gradient here shows you the predicted connectance for any given level of temperature and phosphate levels. And low here means low expected connectance, whereas red means high expected connectance. And then you have the dots, they show you the start and the end of the real liquefaction and warming phase. And then the arrow is just the direction of time. Whereas the dashed lines in the plot, they show you where most of the data lies that I used to produce those models. And so what we can see here, if we follow the trajectory of time, is that it seems like connectance increased in Lake Zurich during real liquefaction and when nutrients were stable. But then again, there's this strong decrease when warming accelerated, where we drastically lose network connectance. If we look at that more general, it seems that network connectance responds in a nonlinear way to changes in water temperature and phosphate levels. And we seem to observe the lowest connectance at high temperature. What I also wanted to show you or point your attention to is like if we look at where we are right now, if we would slightly increase the nutrient levels, like saying going from oligotrophic to eutrophic again, we might drastically lose network connectance. So the second graph here is the same, but this time the color code shows you the interaction strength, which is calculated by this cross-map accuracy. So blue, again, here means low predicted interaction strength, and red means high predicted interaction strength. And what we can see here is there's not much change in the interaction strength over real liquefaction and the stabilizing phase. Whereas we've seen the interaction seems to get weaker when warming accelerates in Lake Zurich. So what we also can see here is that we tend to have weaker interactions at higher temperatures. So by seeing that actually temperature and nutrient levels affect a network's connectance and interaction strength, I was then wondering, what about trophic controls? I think trophic controls are important because if a system is more bottom-up controlled, which means resources control their consumers, a lake becomes very sensitive to nutrient inputs. Whereas if a system is more top-down controlled, which means consumers control their resources, if we want to manage a lake's productivity, we might have to do food web manipulation, like restocking of fish. And so the first question is just, again, does trophic control change over time? And so here on the y-axis of this plot, you can see now the difference between links, like top-down links that go from the top of the food web to the bottom, and bottom-up links that go from the bottom to the top of the food web. And so the color here shows, again, the 10 different lakes. And as you can see here, the trophic controls seem to be highly dynamic and flicker a lot more than what we've seen before. And if we look at everything that is below zero, it indicates that we have more bottom-up links in the food web at that given time, and it indicates stronger bottom-up control, whereas everything above 0 indicates that we have stronger top-down control. And we can see that with most of the lake, top-down control or the top-down links are more frequent than bottom-up links. And what we also can see here, if we follow the trajectories of time, it seems that in Lake Zurich, in the warming phase, where we have this drastic decline in connectance, we also see that we lost some top-down links relative to bottom-up links. And so the second graph here on the y-axis is the same, but this time looking at interaction strength. So it's the difference between the strength of top-down links versus the strength of bottom-up links. And as you can see here, it's even more flickering around the 0 line, and it seems that in half of the lakes, bottom-up control is stronger, whereas in the other half of the lake, top-down control is stronger. And then by seeing that they change over time, now it's again the same question. So are trophic controls affected by those changes in water temperature and phosphate levels that we saw before? And so here again, I use the S-maps, which are those locally weighted linear regressions, to predict this time the difference in trophic controls based on a gradient of water temperature and phosphate levels. And so what you can see here from this plot is that everything below 0, so in blue, indicates that the system is more bottom-up controlled, whereas everything above 0 in red indicates that the system is more top-down controlled. And if we follow the direction of time here in Lake Zurich, it seems that we gained more top-down links during reliquefaction and the stabilizing phase, whereas when warming accelerated, we lost those top-down links in proportion to bottom-up links. And what we can see here that we tend to have stronger bottom-up control at high and low temperatures. So the second graph here is the same, but this time the color gradient shows you the difference in strength of the trophic controls. So where again red means stronger top-down control and blue means stronger bottom-up control. And if we look at the trajectories in time, we see that trophic control decreases towards bottom-up control during re-electrification and the stabilizing phase, whereas there's not a lot of change during warming. However, if we look at the plot, we see that at low temperature and low nutrient levels, we seem to have stronger top-down control here, and at high temperature and high nutrient levels, bottom-up control seems to be stronger. However, I want you to note here that predicting trophic controls is very complex. So what I'm showing you is the scale of the model that is not as good as the scale that I had for network connectance and interaction strength. And this can have many reasons. Like one of them, the model was very simple. I just used 2 environmental variables. It could be that, like, for example, the community has an important aspect. And by just like sorting them into 16 guilds, we might lose a lot of information, what is actually driving trophic controls. So yeah, we saw now that warming and changes in nutrient levels can affect ecological networks. But we might not be able to measure the whole network all the time. And so another question that I had was just to take a deeper look in looking which guilds actually are the most important contributors to network structure. And to do so, I just averaged the interaction strength across time, but the average interaction strength between guilds across time, and then also across lakes. And so what I'm going to show you here is an interaction matrix, and where variable 2 is affecting variable 1. And the interaction strength here I calculated by multiplying the average interaction strength by its prevalence. Whereas prevalence, for example, if a link was significant 50% of the time, it would be 0.5. And so the color gradient here shows you the interaction strength. So whereas blue means low predicted or low interaction strength between those guilds, and red is again high interaction strength. And what we can see here is that actually ciliates, which are very small grazers seem to be very important for the ecological networks in those lakes. And they're very beautiful creatures, as you can see in this video. It's like a master's student took this. And I just want to note that salients might be very important, and we should start monitoring them more closely, because like in the 10 lakes that I had, only 4 of the lakes had data on small grazers. So with this, I already want to conclude my talk, and I hope that I have showed you in the past 20 minutes that ecological networks are actually highly dynamic, and they respond in a nonlinear way to changes in their environment, like your water temperature and nutrient levels. Moreover, some of the lakes in the study might be now, after being warmed, in an unstable state, where a slight increase or decrease in nutrients can have devastating consequences for the network. But also, I hope I have showed you that with the adequate data, ecological forecasting actually becomes feasible, and I hope that the tools presented here can then be applied by researchers and stakeholders alike. So yeah, with this slide, I want to thank, first of all, SEAL, the International Society of Nemology, for awarding me with this prize and letting me enjoy such a great conference. It was wonderful to see all the great research that is produced here every day. Yeah, and then I want to thank my co-authors and also Marta, who is our plankton taxonomist, for her knowledge. And then, of course, all the people that contributed to this amazing data sets that I could use for my study. So thank you.